Well, thank you, Madeline. Thank you for inviting me.
Yeah.
It's great to be back here on campus. I too am, like Dr. Schilling, I'm kind of a Rutgers guy. I started my undergraduate work down on the Cook campus, 1991. I had the pleasure of taking courses here on Bush. I love this campus. On that ride over, occasionally we'd hit the golf course, and at that time, the green fees, I think, were $6 for students. It was wonderful. In fact, I may have hit the physics building a couple times, but we didn't tell anybody that at the time. I came back from my graduate work with mosquitoes down the entomology building, and then I ended up in the Hunterdon County Health Department. I've been there ever since, so it's been about, 19 years now. And I'm a mosquito guy by training. That was my thing, mosquitoes and black flies. Over the years, we've worked very closely with our Master Gardeners group. I've worked with Rebecca Magron in particular in recent years. And I'm trying to think of who her predecessor was. Oh, Martha Milletta, a very nice woman. Some of you may know her in the audience, and she's doing quite well, as I understand. And over the years, as Dr. Schilling alluded to, you and others around us in the health department have been the eyes and ears of what goes on in the community. And it's not uncommon for an insect to come in and wonder what it is. And this is more storytelling than it probably is science today. But we have an example of that, and I think it illustrates just how important it is to be suspicious. And if you are, there's probably good reason. Don't ever think for a second in the biological world that things are stagnant. And nowadays, especially with the way people travel, we're getting bugs in and out all the time, whether it's on animals or people or otherwise. So I'm going to give you an introduction to this tick that we call Haemaphysalis longicornis and its discovery in Hunterdon. And there's a role that a lot of people play in this. I'm going to highlight some of those. I hope I don't skip anybody. So an interesting story. So August 2017, it was August 1st actually, I had two offices at the time. One was in our health department. I should mention I'm a health officer by training as well. So I'm up in our health department, and then I've got our mosquito building on the other end of campus. And my assistant in the mosquito building called me about 10 in the morning. He said, Todd, there's a resident here. She says she knows of you, and she brought in a few ticks. What she didn't know is she was covered in ticks. And she had been shearing her sheep, thought she had a few ticks on her. It turns out they were larvae, and they were these larvae right here. What she didn't know at the time was she was covered, so as my assistant gets to meet her, he realizes her entire clothing is covered in ticks. And his comments on the phone, and I should mention in context, this is at the point where there is a lot of salacious material in the media about inappropriate interactions with corporate executives and this and that. And he says to me, he said, she's freaking out, we're kind of freaking out, and her pants are in the freezer. And I just kind of shrugged in my chair, like, ugh. And he's kind of an excitable guy. He said, but she had to change her clothing. I said, well, next time, mention that first. And in true fashion, like any good entomologist would know, there's two easy ways to kill bugs. One is to freeze them, and the other way is to heat them. So we could have stuck stuff in the dryer down in the basement there, or he decided to put them in the Revco, which was a good move. And she happened to have a change of clothes. So, and to this day, by the way, I just got reminded of this last week. This is, 2 years ago now. Her pants are still down in our lab. She didn't want them back. So went down there. I didn't expect to see anything, to be honest with you, because if you know the tick world, come July and August, what do we see? Are larval deer ticks. And I should mention up front that the neat thing about ticks is the larvae all have 6 legs. They do not have 8 legs. They get that. It's always been a fascinating thing to me, and I know Alvaro probably knows a lot more of the reasons why for this, but they gain 2 legs when they molt, which I think is really neat. Somebody asked me not long ago, which 2 legs do they get, and I don't know the answer to that. But they gain 2, and they end up with 4 on each side. They're bilaterally symmetrical. So I took her pants out of the freezer, I shook them into some pans on the floor, and they're small enough for that I thought they could have been mites, actually. And we do see occasions where people run into a nest of bird mites. In fact, we had an interesting case years ago where a young schoolgirl, I don't know, 3rd grade, something like that, grabbed the ball out of the gym room. They were playing ball. There had been starlings nesting in the walls of this room, and these bird mites had migrated off, northern fowl mite, covered these balls, and the next thing you know, she had mites all over her. She goes to class. She's the one with cooties. The parents are going crazy. The principal thinks she brought them from home from her chickens, and on and on and on. So it's not uncommon to see these sort of things. What struck me as odd when I put them under the microscope, that in fact they were ticks. And the reason we know that is, among other reasons, is, and it's hard to see here, you've got the scutum here, and you do not see that with mites. Not to mention they only have 6 legs and mites would have 8. So, so my initial instinct was, well, this is the time that we would see larval black-legged ticks, deer ticks, right around mid-July to August. And the theme with larval deer ticks when they come out is just imagine you've had 2,000 eggs hatch out all on a stalk of a plant. We see this all the time. A child runs through a playground. What do they get? 50, 60, 70 larval deer ticks on them. And the neat thing about larval deer ticks is they don't last. They start to bite and they realize you're not the right host and they drop off. Of course, it scares the parents and they napalm the house and all that kind of thing. But that's not the most uncommon thing in the world to see. So when I got this phone call at 10 in the morning, I figured I'll be done at the end of the day. I expected to find those. And the one thing I knew when I looked at these on the scope was those are not the ones. Now, I should mention I'm not really a tick expert. I do ticks by default like you do with a lot of bugs. People bring them to you, they expect you to know everything about them, and you kind of shake your head and probably look at your neighbor in the office wondering what to do. That's usually when I get a call from Rebecca or I call her in Hunterdon County. So what we're most familiar with are these guys right here. Black-legged tick, otherwise known as a deer tick. This is what drives 95% of the Lyme disease cases, not only in the state of New Jersey, 95% of the Lyme disease cases now, I should say. It's almost 100% in Lyme disease cases, but between mid-May and, say, the 4th of July, 95% of those Lyme cases will occur in the state of New Jersey and Hunterdon County on the northeastern seaboard. And the reason is that the nymphs are present. But what happens, and ticks are on kind of a 2-year cycle, is the next tick you're going to see is actually the larval deer tick, and that's the one that shows up mid-July to August. And those are the ones that are out in droves, although these are the driving force in the tick-borne disease cycle, not just Lyme disease, but things like anaplasmosis and babesiosis. The other common tick, and we do not see this a lot in Hunterdon County, but I grew up in the Pine Barrens of South Jersey, and this is a tick that we'd always get on ourselves as kids. And to this day, I think it's a more prevalent tick, although we now see it in Hunterdon, and this is the Lone Star tick. If you look, the obvious thing you'll probably notice here is the shapes are very different. And this is true to form. You can see this with the naked eye. You don't need a microscope. And if we go back to that larval tick, it's got that roundish shape. So I thought, oh, perhaps it's a larval Lone Star tick, which I've never first of all, I've never seen those in my life. But the interesting part is there's things that are weird here, and you might not notice them right away, and we'll get into this a little bit more in a little while, but these mouth parts are very stubby. Lone Star ticks notorious have very long mouth parts. It's very obvious to see on the microscope. You can actually see it in the field. And if you look here, rather lengthy in size. So I knew it wasn't a Lone Star tick. The other big one that we probably all grew up with is what we call the American dog tick or the wood tick. Those are synonyms. And I should say there is a brown dog tick, not to be confused with that different genus. That's the parasite of dogs. But this is our most common tick probably across the board outside of probably now the black-legged tick. And the other interesting thing about this is I've never seen the larvae of this. I've never seen the nymphs of this. The reason I've never seen the larvae of this is they actually live underground. They live in dens of things like moles and voles and that sort of thing. They take a blood meal, and you generally do not see these until they're adults. So what's kind of odd or interesting is with the deer tick, we see everything. We see the nymphs, we see larvae, we see the adults at specific cohorts throughout the year, pretty well defined. It so happens this year, by the way, is a very low year for deer ticks, and we can't figure out why, and it's not just us. Lone Star ticks, yes, you'll see the nymphs in droves in the Pine Barrens. They kind of extend up off the coastal plain, and we're now seeing those in Hunterdon, and that's a recent phenomenon. They've been moving in recent years. We always see the adults, never see the larvae. And then in the case of the dog tick, really the adults about the only thing we see. And those have been in very high numbers in recent years, crossing over into weird habitats such as deer tick habitat, which is kind of a fairly new phenomenon in the last few years. So what I knew was it wasn't these. Now there are some very odd ticks that I've never seen before. And to fast forward a little bit, one of those is in the genus Haemaphysalis. It so happens that genus, and we'll show you some pictures of those ticks, is a very easy genus to identify. Getting it to the species level is a little bit tricky, at least now that we have some introductions here. But getting it to the genus level is a piece of cake. It's like, anybody ever use dichotomous keys here to ID insects? Okay, so just for those who haven't, when you ID insects outside of just visually looking at them or genetically looking at them, the traditional way to do this is through a dichotomous key. And what it is you have two choices. If you choose A, you go to this part. If you choose B, you go here. And every time you have two choices, The problem with dichotomous keys is you will always get an answer, regardless of whether the species is native or not. So what you need to do is when you get an answer, you have to ask yourself, does it make sense? And where we'll go with this is we had an answer, a very obvious answer, but it didn't make any sense based on the biology and some weird things about the tick. So I just wanted to give that background that we see this tick now. It looked like nothing I've ever seen before, but it is very obvious in this genus. And it so happens we have two Haemaphysalis ticks known on the, let's call it east of the Mississippi River. One of those feeds on rabbits, and rabbits only. And the other is a little weird. Nobody knows much about it. Probably birds, that sort of thing. And that's it. That's it. But around the world, there are lots of species, especially once you get into Australia and East Asia and that sort of thing. And that's where this story ends up leading us. So, as I said, right now, this is what we'd expect to see. This is what you're seeing out in the environment, just so you know. This is the nymphal Ixodes scapularis. The paradox of these ticks, just so you know, about 1 in 5 of these has Lyme disease in it, yet they transmit 95% of Lyme cases we'll see this year. The adult Ixodes scapularis, about half of those have Lyme disease in them, yet they produce very few cases. A few reasons for this. One is they're very delicate biters. You generally don't feel them until after the bacteria's entered your body. And the other thing is they tend to be more concentrated than the adults. So if you get one of these on you, you probably have 2 or 3 in a lot of cases, whereas adult deer ticks are very spread out. Maybe you get one on you, you feel it before it transmits disease. So I just want to be aware that's what we're seeing in the environment today. And as you can see, it's got that, even the nymph has this teardrop shape to it. And these Longicornis ticks are rather spherical in nature. Okay, so the lady came in August 1st, and she was a little bit, I won't say odd, but a little difficult to communicate with. Her cell phone, the voicemail was full, so we couldn't get in touch with her there. She did not respond to emails. So we basically did nothing. We didn't know where this tick came from. I didn't really have any reason to be concerned because it never occurred to me it could be an exotic. So about 2 weeks later, she came back, and this time I was at my mosquito desk, and this is what she brought in. And I'm just gonna pass these around. I just want you to get a sense of the sizes of these. So you can just pass those around the room. I'll grab them at the end. These are, this is a family portrait of Haemaphysalis longicornis. There's your larva. That's your engorged nymph. And this is your engorged adult. And I know Alvaro probably knows more about this than I do, but you know, we're talking in the thousands of eggs here that adult's going to produce. So if you can just imagine the dynamics here, if one adult female lays eggs, you get a lot of ticks at the end. What's also interesting, and this story's yet to be told, but this tick, unlike any other tick I've ever heard of, or for that matter, most arthropods, can reproduce through cloning. So if it gets stressed, or maybe it doesn't get stressed, instead of having to mate with a male, it just keeps cranking out babies. And the reason I mention this is it goes to explaining the sheer numbers that we saw. And we were at a point in the cycle we didn't know where we were. But just so you know, where we are today is we're seeing nymphal Haemaphysalis longicornis right now. We're seeing adults as well. Those adults are going to produce eggs. We're now in June. Come August, those eggs are going to be hatching, and those are the thousands of ticks you're going to see, and that's what she brought in and didn't know it. So in any event, she comes into our office, and this time she had taken ticks off her pet sheep, and this is what she found on her sheep. So I didn't have a chance to look at them at that time. I said, look, I don't know what the last ones were. I don't even know if these are the same, but I'll take a look at them. We'll get back to you. And we could almost always get an answer. It may take a little while. Well, I looked at these, and I had no idea what they were. And just so you know, most dichotomous keys are based on the adult arthropod or insect. So it should be easy to figure this out, and it was not. Nothing made sense in the morphology of this tick. So I sheepishly, no pun intended, said to her, I have no idea what these are. I'm embarrassed to admit that. In fact, I'm not really a tick guy. I don't even really know who to ask. And I thought about it. So this went on for the better part of a month. Now, there's a nice tick expert in Monmouth County. His name is Bob Jordan. I sent him some. It turns out he never had a chance to look at the samples. But fast forward about a month, and I was at a meeting with, it was a vector-borne working group. It's a meeting that meets throughout the state. I occasionally go. It meets once a month in the summertime. And I mentioned this story, and there's a geneticist there who I got to know. Her name is Andrea Jazizi, and there she is. I have a picture of her. This was the break in the case. So a few things happened in between. I'm trying to get my timeline straight. But anyway, I spoke to Andrea and I said, look, I've got this tick. I don't know what it is. I'm not even sure where to go. I touched base with a few people, one including a gentleman at the Smithsonian who's a tick expert. And his comments were very interesting. He said, I don't know exactly what this is either, and he's a tick expert, but he said, I can tell you it's not from North America. And that's when we knew we had something odd. So she just listens to this conversation. She's a geneticist. And unbeknownst to me, she went back to her office in Monmouth County. Now, she's got an appointment at Rutgers, I should say. Went back to her office in Monmouth, grabbed that sample that hadn't been looked at, and ran some genetics analysis on it. And it came up with about 99.9% certainty that it was this Haemaphysalis longicornis. Now, usually those tests come back, or 90% certain it's somewhere in this lineage. This one came back almost a sure fit. So we quickly researched that tick, and it was funny because after I had identified it as a Haemaphysalis tick, I just Googled it like everybody else does. And sure enough, everything that kept popping up was Haemaphysalis longicornis. It's a real problem around the world. And I always discarded that information, even though it looked a lot like it, just because this was found in New Zealand. It had no business being here. I couldn't explain why it would be here. It just didn't make any sense. So at that point, what we did, a few things. It triggered a whole series of events. One of the things was I reached out to, and I should say, this was confirmed morphologically then at the National Veterinary Services Laboratories. Now, these NVSL labs, if you know anything about them, that is the world premier lab for anything animal related. Second to none. I mean, they work in concert with CDC, but this is the animal end of that. They don't make mistakes. They're very selective about what they take in, but when they take it in, it's important. And they confirmed overnight, and it turns out they had an expert on the Haemaphysalis longicornis who works there, has been there forever, and he knew exactly what they were as soon as they got them. But in the meantime, a couple other people, Rutgers folks, played an important role in this. And I figured, I'm at Rutgers, not only should I mention them, I should highlight them, just so you're aware. Dr. Dina Fonseca, who's the director now of the Center for Vector Biology, we've known each other for, I guess, 20 years now. I don't know her personally, but just know to interact with her quite a bit. Her student, her part-time student at the time, Jim Ossie, who's now at our state labs, I'd talked to him because I knew he knew a lot about ticks, and he didn't know what they were. But he immediately, referred it to his boss. And we got on the phone together, and she said, we've got to notify somebody about this, but I don't know who. And I know that's one of your challenges. You get calls in, you're not really sure where to go with them. And she said, here's what I'm going to do. I'm going to reach out to CDC and USDA. And sure enough, they were both interested because this tick has huge implications for livestock and animals, wildlife. It also has some implications for human health. There's some weird diseases it transmits overseas. So this triggered a series of events, and I kind of put the cart before the horse. But what happened is the USDA folks immediately got on the phone with me, and this is now November, and they said, we need to see these ticks. And I figured, okay, I'll mail them out to you, whatever. And they said, no, we need to see them like now. And I still remember this. This was election day. It was Monday, before election day 2017, maybe November 6th, somewhere in there. I said, look, we're closed tomorrow, which was not an acceptable answer to them. They're the ones who get shut down every now and then, by the way, and nobody ever says boo. I tease about that. But they said, we need to see them right away. I said, I'll tell you what, I've got my 4-year-old son at home because my wife's working. But if you'd like, I'm more than happy to come in as long as you can put up with him for a little while. I can have whatever you'd like. Sure enough, they sent somebody out. She was there the next morning and gave her all the ticks I had. And that's when they went to NVSL overnight. Within a day, I had an answer of what they were. They FedExed them overnight. And sure enough, they're Haemaphysalis longicornis. So I just want to acknowledge them because they did, Dina and Jim did a few really important things. One is Dina connected us to CDC and NVSL. And the other important thing that I think is significant for you as well is she conducted an extensive literature review on Haemaphysalis longicornis. And this is a big deal because I'm not in the academic world. I don't have access to the latest literature or how to get it. But the faculty, at Rutgers and other universities do. And she was able to uncover some amazing things. I mean, she was finding theses of work done in China on this tick. It was kind of funny because some of the abstracts were in English, but the rest was not. And we're trying to sift through that. And I had somebody who spoke Mandarin in our nurse's office, and she helped me understand some things. It turns out this tick goes from, let's call it New Zealand, right through South Korea. And there's actually an American researcher who's been working on this tick in South Korea and had a lot of insight. So that was very important. What was fascinating about that was, since this tick is found in the southern hemisphere, I didn't put the pieces together. Nothing makes sense at first. But they're finding this tick in February or whatever. I'm like, this doesn't make any sense. It turns out it made all sense in the world. Everything's the opposite down there. That's their summer, and this is exactly when we should see it. So it turns out, as the story goes, just to fast forward, the biology overseas is exactly the biology here in the United States, which is interesting because you never know how that's going to pan out. Jim Ossie, I sort of referenced, he connected us to this gentleman, Richard Robbins, at the Smithsonian. Now, it turns out Richard Robbins is now semi-retired, and it was fading into that at the time. He knew everything about ticks. When he didn't know it, he was very interested because he had never seen this particular species at the time that he knew. Jim also provided us some in-depth background on ticks, so it got us a good sense of where to look. And to this day, we still work together quite a bit on these things. So they played an important role. So as we go through this, I'm just going to mention some people, and then I'll give the acknowledgments at the end officially. So while all this is going on, so this is now November, but let's back up. We connect with this lady. We lose touch with her again, this resident. Her phone doesn't work, doesn't respond to emails. And I'm meeting with a colleague of mine from DEP about a black fly problem we had. That's my other hat I wear, is we do some black fly control. And he's sitting across from me in my office. My phone rang. And for whatever reason, I picked it up. I don't have caller ID. I just picked it up. And it was she on the phone. And she was really funny. And I said, where are you? She said, I'm at the paddock with my sheep. It turns out that's not her house. Her sheep is at this gentleman's house. Now, as the story goes, and this is kind of interesting, relevant to you, this sheep was part of a 4-H project for their kids years ago. They had 5 of them. This was the last remaining one. The others had died. Her kids are now through college. Well, guess who feeds a sheep every day? She does. Guess who does this? Gives her water? She does. And it's kind of interesting. At this point, I knew the health of her sheep could be impacted by this tick, and all these suggestions how to give her the tick. I could just see her looking at me like, kind of almost hoping the sheep might go so she doesn't have to feed this anymore. Because every time I have suggestions, she's like, you don't have to do that. I'm like, no, we really should. So in any event, I said, I got it. So she's on the phone with me. I said, are you going to be around there for a while? She said, yes. I said, where are you? Turns out she was 10 minutes from my office. So I said, don't go anywhere. I said, I'll be there. And I knew exactly where this area was. It turns out it's near a black fly habitat area that we treat all the time. It couldn't have been a mile from there. So I hung up the phone, and I looked at my colleague from DEP, and I said, where do you need to be in the next hour or two? He said, no, I just got to get to my office in Trenton. He was driving through Hunterdon by 1. I said, I'll tell you what, we're going for a ride. Now, this gentleman, I've known him forever. This is one of the handiest guys I know. He's an entomologist by training. He's an electrician as a hobby. I always call him about personal favors more than anything else. It helps him with plumbing or whatever. What I didn't know was he did not like ticks. He had Lyme disease, and he'll crawl in a sewer to fix a pipe, but he does not like ticks. I did not know that. So we go out to this farm. Let's see. And this is it. By the way, this is the sheep Hannah. So this sheep was covered in ticks. Let me just show you that. So we just pulled up an ear. Ticks to the point where they're hard to collect. They were on top of each other. Now, what was interesting, I didn't show you in this picture, is this is 4 inches of wool. It's an Icelandic sheep, which I never heard of, by the way. The ticks are all the way to the skin. I don't know how they got there. But in any event, we go out to the farm, and I see Hannah in the field. It's about an acre paddock. And I said to the lady, the resident, I said, well, I need to get some ticks off of Hannah. And she said, you're not catching Hannah. I said, well, I'm not leaving without the ticks from Hannah. And we had this little debate, like, I'm going to collect these ticks. Oh, no, you're not. So it turns out Hannah didn't like to be caught. So I'm in my steel-toed boots. It's 90 degrees out. This is in September. And next thing you know, I'm chasing the sheep around the paddock for 15 minutes, to the point where I couldn't chase anymore. And what had happened prior to that is my colleague and I from DEP had walked in, and we immediately got covered in larval ticks. And out of nowhere, he's like, "Boop, I'm out." And he goes by the barn. I'm looking at him, I'm like, hold on a second here. I need your help. And he's like, nope, ticks is the one thing I'm not helping you with. So I'm chasing the sheep around. And it's kind of like we were thinking alike. I think we were both about to give up. And out of nowhere, the sheep darted for the gate. So what did I do? The 10-year-old football player came out of me. I ran straight after it, and I tackled the sheep. And it was down, and I did a fist pump, a Tiger Woods fist pump right on the sheep. And then it suddenly occurred to me, it wasn't moving. And I looked up, and this guy from DEP is like this, and the resident's like this. And my first thought was, oh my goodness, the call's coming in the freehold director. I might as well put my 2 weeks notice in now if they'll let me keep that. I killed the sheep. But then it got worse because I could feel it breathing. I'm like, it's worse than I thought. It's paralyzed. It's even worse than I thought. So it turns out Hannah didn't like to be chase, but once you had her, she loved to be coddled and this and that. And we sat there for the next 15 minutes picking ticks off of this thing like it was nobody's business. So we get all these ticks, and so fast forward, we ended up having these meetings with Andrea, and that's when we figured out what it was. So I looked at the resident and I said, look, this sheep it's going to die. I mean, it's old. It's going to die of anemia, if not shock. And she was kind of funny, and she's sort of like, oh, whatever, maybe it will die, or something like that. So happened I had I brought some stuff with me. I always keep a little bit of neat stuff on the shelf. I had an old livestock wash. It was permethrin-based. And I had put her in touch with Bob Mickle, actually, the Rutgers extension at the time. And he really wasn't sure what to do. And so while we're there, I said, look, I've got a permethrin wash. It goes an ounce of a gallon of water. If you'd like, I'll give you a little bit. Turns out it killed everything. It was perfect. Killed all the ticks off the sheep. It was wonderful. In fact, we felt at the time, the gentleman from DEP is a pesticide guy. He said, you know what, Todd, this could be a tick-killing machine. That'll be a nice coat of insecticide. It'll walk around and probably kill the ticks that come to it, like we would when we use permethrin sometimes. You'll see that on your clothing. So that worked quite well, by the way. So fast forwarding to a story not told today, but one of my recommendations to people is when you get a problem like this, we had an acre to deal with. We'll talk about killing ticks in an acre. If you've got a 50-acre farm in Sussex County or something like that, there's not enough money to buy insecticide to do that kind of work. You may end up treating your animals, which is very cheap and easy to do, is kind of my sense of this. But in any event, moving along here. So I'll just keep track of my time here. We started collecting ticks, and for those who aren't aware, there's a few different ways to collect ticks. One is human bait, like we just walked around and that ticks all over us. Or you can dress up in Tyvek and all this kind of, I use waders myself usually, but we use what we call either tick drags, which is a defined area of cloth, and you drag them around depending on the habitat, or more commonly, although we don't see the pole here, I don't think, a tick flag. And basically it's a PVC pipe with fabric on it, and you go around, and that's usually how we collect ticks. So in the industry, if you're looking to collect ticks, those are the two common ways. And Dina Fonseca, by the way, has a nice protocol of exactly how to make this tick flag. They can be very sophisticated with some PVC pipe. We tend to use, I tear up an old shirt, and put it on a piece of wood. It's low budget operation in the wealthiest county in the state, by the way. Just had to throw that in there. And this is what we found. And all these little specks of what appear to be dust, those are larval ticks. Very patchy, like a lot of ticks are, but we would get them 1,000 here, a few here. What was interesting was, and I'll show you pictures of the paddock in a second. So this is September, larvae, no nymphs. I finally found one or two on my shoe as I was walking around. No adults, yet on the sheep, we're seeing everything. So something's not right here, and that we finally figured out what that was in hindsight. And it so happens they utilize, and I thought about this at the time, they utilize different habitat. So the larvae are high up, and we're hitting those with a flag, whereas the nymphs and the adults are low to the ground. In fact, I've yet to see this, and Jim Ossie was working on this a little bit. I've never seen a tick do this before. They overwinter in the soil, not on the surface of the soil like most ticks, which are, by the way, in a state of torpor. They don't really hibernate. But these actually go underground and survive the winter, which is fascinating. And we tried taking some soils. Jim was out there with, those devices you use to cut a hole in the green on a golf course. He was out there trying to take some soil logs. And I don't know if you ever found them over the winter, but that's in the literature, and I believe that's where they end up because they sure enough did show up in the next spring. But this is what they look like. So just a little bit about tick morphology, and I know we'll have a lot more expertise with Dina Fonseca later in the day. So what makes this weird or different? A few things. When you're looking at a tick, and this is valuable to know just if a resident calls in, first of all, we've got this important part here. This is the scutum of the tick. They all have it. But the females, it's small, relatively speaking. I didn't put a picture in here, but on the male tick, that encompasses just about the entire dorsum. And so male ticks look kind of weird, and that's why. Is that sclerotized region of exoskeleton goes all the way down. Female ticks, it's always some sort of shape like this. Now, sometimes it's trapezoidal, sometimes it's a little bit circular, and that it can go towards identifying a species, knowing how that looks, basically. Is it mottled? For example, if you can see, as you can see with this one, there's no color. You probably know this, but when you get a wood tick on, you can immediately see that white mottling on the scutum. So that's somewhat unique, not necessarily species specific, but it will get you to the right place in a key. The other thing, a little bit hard to see on this picture, you've got these ridges down here. Those are festoons. That's not terribly common in ticks. We do see that, but we certainly don't see that in black-legged ticks. And you'll see these in the larvae as well. So you see them larval, nymph, and adult. And by the way, I should mention, with larval or immature arthropods, they lose and gain characters as they grow. We talked about ticks gaining 2 legs. Mosquitoes gain hairs and lose hairs all the time. So it's very dangerous. I said dangerous is probably a strong word. It's risky to identify something that's not an adult if you're not familiar with the species. And I've fallen for that before, and I still do to this day, and I keep reminding myself of that. So the other thing I mentioned is these stubby mouth parts, and that's a relative term. You wouldn't know they're stubby unless you saw long ones, but if you ever see a deer tick, they're quite large, quite long. What you can't really see in this photo, but this is what really had me scratching my head, was inside of the sclerotized area, or the palps. That's what enters into the skin. On the 2 species of Haemaphysalis we have in North America, the dentition is very simple. You can actually count this symmetrical dentition, and it's either 3 on each side or 4 or 5. So you'll see it in the key written as 3 plus 3, which means 3 on this side, 3 on that side, 5 plus 5. This one had too many to count. I didn't know what I was seeing at first, but it turns out there's so many, they're so small you couldn't count them with microscopy at about 40 to 80 magnification. So that was just a clue that something didn't make sense here. If we flip it over, so now we're looking at the ventral side, the underneath. You can see this dentition a little bit more clearly. They look like little bumps there. A few things about ticks, just so you know. This is the rectal area here. There's a ridge that goes under here. Whether or not this encompasses the rectum or not, and this one does not, is diagnostic to ticks, and you'll see that in the key. So that's something important to know. You can really see these festoon ridges here. There's also some things you can't see very clearly on the slide, but that triggered me. Now the other weird thing, and I think I have a slide of this next, is going to be right here in the mouth parts. This is what had me thinking this is just something that's I don't know. Let's see, yeah, I do have it here, a little zoomed in area. So right here, they're like fangs. They look like fangs, what they look like. I think that's where it gets its name, longhorned tick. These projections are not seen in our native Haemaphysalis. They're just not there. So I've yet to actually collect a rabbit tick on a rabbit, although they exist. I've seen them in collections. But you will not find those, even though the tick itself looks very close to our native tick. So they can be easily misidentified. And we see this all the time, where residents bring samples in, we misidentify them. And usually it's not a big deal, and I'm not saying it is a big deal in this case, other than we happen to stumble across something that's now invasive across half of the United States within the last 2 years. So just getting back to a little bit of the field work. So we brought in a bunch of people, and these people are a little bit nervous, so they've got their gloves on, their Tyveks. This is one of our, very nice, one of our state vets. She was kind enough to bleed the sheep, and the reason she did was she wanted to see if the ticks gave anything to the sheep, any weird diseases. Now, in the world of testing for diseases, that's a complicated business, and we'd like to simplify it, saying we have Lyme disease or this. Well, it turns out there's a lot of stuff in ticks. Traditionally, what we do is we target a pathogen, a bacteria, whatever, and look for it with a PCR test. Now, the more recent sort of phenomenon is you do this genetic shotgun approach, where you sequence everything if you can. That technology's there now, because it turns out there's some really weird stuff that these ticks transmit overseas that we just don't have a lot of familiarity with over here. Now, that story's yet to be told, and there's people at Rutgers working on that right now, and I'm sure other places. But in any event, this is the field, you see a little perspective. So what made this tick kind of unusual compared to what we usually see is this tick was out in the open. Now, normally we're talking about deer ticks. They're in your transitional area where you go from this field to the woods, where that grass is shaded, they're low to the ground. This tick was high up in the weeds here. This is just basically an unkempt paddock where the sheep ran around, and the larval ticks were everywhere. Now, like I said, what we didn't know at the time was, and we sort of suspected was, the other ticks were a bit lower, and they probably come up and they feed when they can, but they stay out of view a little bit. So we decided, so we got on the phone with USDA, and I think they want to napalm the place. I really think they want to quarantine the area. It was funny because, not that we're any different in terms of home rule in Hunterdon County or the rest of the state, but when you talk about quarantining somebody's property, red flags go up all over the place. We just don't do that in New Jersey. Certainly not in the year 2019. Maybe in the year 70 or something, but not now. So we convinced the USDA. First of all, we didn't know what we had. We thought maybe it's isolated. Turns out it was not. But we felt we could, what we knew was we did not find these ticks outside of this paddock. We sampled in the woods, couldn't find anything. So we thought we had it fairly contained. So our thought was perhaps we can kill the ticks in the paddock. So we quickly recalibrated all our mosquito equipment. to kill ticks. And what's interesting in the mosquito world is we use very little insecticide. We'll treat on an acre, we'll use maybe an ounce or two of malathion, an ounce or two of pyrethroid. In the tick business, we're looking at 50 ounces. So our machinery was not calibrated for that. So we had to start from scratch, and it doesn't take long other than, of course, the sprayer we wanted to use, we haven't used in 10 years. The carburetor's gunked up. We had to get that replaced. And then these carburetors are this big, and my fingers are this big. And so we had our mechanics help us out. So what we decided to do was, we felt we had ticks below the surface, which we did. So we figured is let's do some habitat modification really quickly. And that's what I recommend too with deer tick control is you want to clean that habitat up if you can to one, perhaps kill some ticks because they won't like it. And the other thing is to make it accessible to your insecticide. So what we did was we hooked up an ATV we had with a rough cut mower, basically a brush hog, and we run around and did just that, and it worked quite well. So we didn't know what to do with the clippings. We've got 2 feet of grass that we're cutting off here, and we didn't want to relocate those outside the paddock. If this were very remote, and if this were Texas, we would have burned it. We literally lit it on fire. But you can't do that in New Jersey, certainly not without catching other things on fire because everybody lives so closely together. What we decided to do was just leave those clippings there and spray them. And that's what we did. So we cleaned up the habitat. And it was not easy. Some of it had multiple rows, which is just a nightmare to deal with, whether it's walking through it or just trying to collect in it. So we mowed down all the briars, got it cleaned up, and then we considered what to use. Now, and I just wanted to mention this just because I know you have residents who call in and saying, okay, I got a bug, what can I kill it with? And I think your policy is, we'll give you some guidance, but we can't recommend products to you. Is that correct? I know that's tip of the much. Okay, well we can. So I told Rebecca Magrin in our areas, when you get those calls, send them to us. We'll tell them exactly what to use. We'll tell them what works, what doesn't, and we're not advertising necessarily for a product, but we'll give them some options there, and that's what we tend to do. But when we do that, what is useful perhaps for you to know is we want to consider safety. You know, we're looking at exposure to people, to other animals, to fish. If you look at a pesticide label, do not spray within so many feet of a stream. You'll see those sort of things. Product chemistry is a big deal, and particularly nowadays with pesticide resistance. Pyrethroid insecticide resistance is a huge problem, especially in the tick world. And we don't do a lot of tick control, so luckily it didn't affect us too much. Then we get into the specifics here. Big differences about how a liquid versus a granular is going to behave. If we spray a liquid on that grass, it's not going anywhere. Now maybe we don't want it to go anywhere, but we knew we had ticks below the surface too. The beauty of a granular is gravity is going to take it down, right? It's going to take it right to the bottom. Turns out we did both. And then the timing of the treatments. And these are things that we didn't know at the time. It turns out it's a little bit more flexible with this tick because it's here all the time. With deer ticks right now, you're killing them now. If you wait a month, you're going to miss your brood, right? So timing's important. You might as well wait till the fall. And then, of course, this all comes down to its cost. We don't have a lot of money. We had a little bit of money to get by. It turns out USDA provided some insecticide, and that was part of the arrangement worked out was I said, look, I'll dedicate my crew to this. Would you be kind enough to provide the insecticide? And that's exactly what they did. It was a part of the theme of this discussion is it was a good example of various agencies working together. And we don't always see that. Usually somebody doesn't want to work with somebody. You're like 2 kids in a playground who don't want to share toys. This was just the opposite of that. USDA jumped in, we pitched in, we had all the in-kind support, and we were able to get a lot done with almost no money, at least to start out. Then, when deciding on a product, as I mentioned, product chemistry comes into play. It turns out in the tick world a little bit different, and Alvaro and I were just talking offline about an IPM program for ticks. We don't have that. We have that for mosquito control. We don't have that for ticks. You've got a few options. Pyrethroids, which I'm not a huge fan of for a lot of reasons. I just don't think they work very well is really the crux of that. Carbamates, which work great, we don't have much of them. Organophosphates are related, act similar to carbamates, we just don't have a lot of those for tick control. A lot of the natural products simply do not work. Cedar oil, all those, they're mostly gimmicks. There are some neat sort of fungal pathogens, that sort of things that are making it to market, but they're just not readily available to the consumer, and we couldn't even get our hands on them. And then the timing of treatments, which historically, May to June, we're looking at nymphal ticks, September, October adults. That didn't apply here, it turns out, which was important to know. So, just a little bit about these. I'll wind up this talk shortly here. Some of the advantages. So with granular products, let's talk granular. The beauty of these is, and this is a big deal today, is less restriction on pollinating insects. We can put these granular products down. They simply are not exposed to honeybees and the like. That's a beautiful thing. less restrictions on grazing animals. It turns out some of our liquid pyrethroids do not use in a pasture with grazing animals. Was this a pasture? We're not sure. We call it a paddock. It turns out what we did is we did use a liquid and then we isolated the sheep elsewhere. So that's how we solved that problem. But if you had a lot of animals, that might not be an option. Beauty of granular products is less restrictions for proximity to water. So a lot of your granular products, since they're not being washed out easily, you can put them, if you have to, really close to a stream bed. In this case, didn't really matter because we didn't have a problem with water. And we need very little equipment. We have these big leaf blower-like things with tanks on them. We blow the granulars out. You've got these things you see on golf courses, which blow leaves. You can use those. You can modify those. So we had the equipment we needed, and it's not very expensive. Now, if you had a big area, helicopter applications are an option, and USDA did explore that. It became unreasonable because it's so expensive. You have to use a lot of product. The disadvantage, of course, of granulars is there's some labor involved. We got guys on the ground schlepping around 40 pounds on their back, especially when it's 90 degrees out. That's not fun. It turns out we're doing this now in November, so it wasn't that big of a deal. So it's more labor intensive. And it's more expensive on a large scale. The granular is mostly tied up with inert ingredients, in this case, corn cob. We've got this insecticide coated either on paper or on corn cob. And that gets expensive because the liquids are much more focused. You're using mostly insecticide there, even in small amounts. So that's the disadvantage. The liquids, let's talk about those. Just so you know, the advantage, we can cover large areas with little product. Less expensive, so you get a big bang for your buck, less labor intensive. We got a nice power sprayer, which I'll show you. Hit the trigger, you go. It's pretty easy. Disadvantage, you're going to need the equipment. The equipment's got to work. Restrictions on pollinating insects, you're going to see a lot of that on these labels, especially pyrethroids. And then you need to use whatever you mix. If you mix up 100 gallons, you got to use it because you got to do something with it. So you got to know exactly how much you need. We decided to go with a couple things. This granular 7, which had 8% Carbaryl. There is one on the market with 2%. We use that as well. This gives a big bang for our buck. We also, I cut and pasted this in, and I forgot to change the title. This should be Demand CS. That's what this is. This is a liquid pyrethroid, so we use that as well. And just a few slides of that. So we've got our hose, about 300 foot of hose on a nice little ATV sprayer that we put in the bed of a truck. It was very accessible. Pour the insecticide. We're mixing that in the tank and then spraying it out in the field. And we killed upwards of 99% of the ticks. We went from tens of thousands. I think we collected 2 ticks there last year. Now, if a host were to come in, the sheep is now gone, we remove the sheep, that would drive this cycle a little bit. So it might be different, if we had a lot of animals. Wildlife were playing an interesting role here. They're moving ticks around, but they're not really driving huge numbers of them, it appears. Just to give you an idea, this is a site. So here's the house, here's the shed, and this is our paddock right here. So within that acre, about an acre, that's where we had the ticks. That's a scale right there. What we did is we literally pulled our truck up here and we sprayed the whole thing. Went in there and mowed. So that's about the story. I just kind of wanted to give you a perspective on what we found, and then I know a lot of people will elaborate on more details with this tick and other ticks. Just wanted to thank a bunch of people. This is my mosquito crew here. Nick Separullo, Sabrina Heron, and Gary Donato, who's a Cook College graduate as well. He and I have been together for about 19 years now. All the people, some of the people I mentioned before in alphabetical order, so not by importance, but Andrea Zizi certainly played an important role. Anna Welsh and Leslie Serafin from USDA, they're the ones who came, and I think you'll hear from Leslie later today. Anna Welsh was the one who came, pick up the ticks, and sent them right to Ames, Iowa. Bob Silva, Chris, and Dan, they're the Hunterdon mechanics. They helped us replace this carburetor on a moment's notice, which is not easy to do, and got our equipment up and running. Brian Smith is one of the vendors who helped find a product that was compatible for us. He's a very nice gentleman. Jack Slater and James Merton, I've never met them in person on a lot of conference calls. They're the experts on this tick in Ames, Iowa. An extraordinarily talented group out there at the MVSL labs for USDA. They work for USDA. Jim Ostie and Dina Fonseca, who I've mentioned, workers in entomology. I know Dina will be here today. I don't know if Jim's here. He currently works full-time at the Health Department labs in Trenton. Has been a real asset to us on the mosquito front as well. Nicole Lewis, who I still keep in touch with quite a bit. She's our local Department of Ag representative, and she helps us facilitate getting ticks to and from areas. Richard Robbins, Smithsonian Institute, he's the one who initially told us we got something weird here. Very nice gentleman, we still keep in touch. And then finally, Scott Kranz, he's the one from the DEP who helped me out with insecticide recommendations, and also was kind enough to at least go out to the field with me and take a look. So anyway, that's the story of Haemaphysalis longicornis. Thanks so much for your attention. I really appreciate it. And if we have time for questions, I'll take the questions. Oh, So right now, the genetics of the tick are being looked at pretty closely. It looks like probably that New Zealand area. To find that out, Andrea is doing this work. She's got to get reference collections from all over the world to compare them. That's been very difficult to do. So she's been working on that, but leaning towards that area of the world.
And where did, how did it
Get here? Oh, should have, yeah, good question. James Merton and Jack Schlater at USDA said, we've seen this tick imported before, it's never stayed, as most recently as 1969 or something like that, came in on a horse. Hunterdon's horse country, which I didn't know is, we've also got a huge sheep trade. I did not know that until this incident. We think probably large animal, more than likely large animal, that's what we think. All speculative though. Yep?
Have you, since August of 2017, has any other place in New Jersey experienced this?
So, after we found it, people started looking through reference collections. Otherwise, it turns out it's been in the country for 10 years, at least. In fact, NVSL had a collection that they had misidentified from 10 years ago. We now have it in 107 areas. And so you've heard of this tick is spreading. Although it does move around, I think it's been in a lot of areas we didn't know it. It looks like, by the way, sort of the moral of the story is, if you get this perfect storm of events, you've got a good host like a sheep and this tick, it takes off. If you don't, it appears to reproduce but not do a whole lot. And we've now found it a few places in Hunterdon. It's been found up in Bergen. It's been found on campus, on the Cook campus, with little fanfares. So it's around, it's probably been around. The genetics though show that there's been at least 3 introductions to the US so far. And ours is different than the one up in New York State, in Bergen County, I believe, as well. So complicated story, but it is around New Jersey elsewhere. Don't know. So it's got, in Asia, it's got a very bad virus, one of those viruses that'll, I don't know, cause your ears to bleed sort of thing. We have not seen that in the ticks here. In fact, we have seen very little other than residual sort of stuff. So that's ongoing work. But so far, no pathogens found, but it's got the potential overseas to transmit. And Alvera could speak this probably better than I can. Some of these bacterial agents like we see around here.
Hannah, one question. Did Hannah survive?
She actually has since died, I say of natural causes. Yeah, she died recently. So Hannah's no longer with us.
But not of the tick problem.
Not of tick, and not of broken bones, apparently, from being tackled. So I'm sticking to that story.
I have a question. Does that tick only spread on those kind of animals? It doesn't, if it gets up in deer or something?
It's been found on wildlife, yeah. So, and it's been found, Vera, again, probably knows more about this than I do. It's been found on small mammals. So USDA sent out a crew from Georgia, and they were trapping everything under the sun, mesomammals as they call them, the small stuff, deer, and it's been found on several things now. And it's likely being moved around by wildlife at this point, more so than people, and probably livestock even. Thank you.